already started talking. One of the most interesting questions I think in the field of speciation is how reproductive isolation varies evolve. And I'm especially interested in the evolution of pre-zygotic reproductive isolation, and especially pre-mating isolation. And usually the first two areas evolve, especially in speciation processes where symmetry plays a role. And I argue that the organism I'm working with, the orchids, are a very good system to study the evolution of pre-mating reproductive isolation, and I'm going to tell you why now. So there are about 200 species of orchids, and males of all these species collect volatile chemical substances from their environment throughout their whole life. And these chemical substances they store in especially adapted pouches on their hind tibia that function as containers for these volatile chemicals. So over time, a complex chemical blend accumulates, and this blend is what we call perfume. Eventually, orchid females release this perfume in order to attract conspecific females in order to find mate. So this very interesting natural history of orchid bees led to a bunch of studies focusing on the chemical ecology of these perfume phenotypes. And today we know that these perfume phenotypes are highly species specific, as you can see here on the lower right in the heat map. And they evolve under strong divergence selective pressures. Also, they're important for pre-mating reproductive isolation. However, what we don't know is what the molecular mechanisms are that underlie the evolution of orchid bee perfume communication systems, and this is what I'm interested in studying during my PhD. So therefore, I'm focusing on two very closely related sibling species, Glossa dilemma and Glossa viridissima, which have a divergence time of only about 150,000 years, which is extremely short in evolutionary timescales. And also, they show a very cool paratype distribution pattern with Glossa viridissima distributed in south eastern Mexico and Alopetri, and Glossa dilemma distributed in Central America and Alopetri, and both species sharing about half of the respective distribution around Yucatan. So these two species are morphologically extremely similar. There's only a single morphological character to distinguish the two species. But when we have a look, if we have a look at the perfume, things are different. Here in this multi-dimensional scaling plot, you see that Glossa dilemma male perfume phenotypes in white and viridissima phenotypes in black cluster by species. And the biggest difference between the two species is a single compound called HNB, of which Glossa dilemma, so the Glossa dilemma perfume consists of about 60% of HNB, but it's completely absent in the Glossa viridissima perfume. So the first obvious question that was asked was whether the olfactory capabilities of the different species are different, and this is the reason why they collect different chemicals for the perfume. So a comparison of the antennal response to chemicals. So the antenna is where the sense of smell in insects is located. And the antenna, as you can see here in the bar diagrams on the right, the antenna response of the Glossa dilemma individuals is significantly higher to HND than those of the Glossa viridissima individuals. And on the left side, you see a chemical 2-odecanone that appears in both species perfumes, and there's no difference in the antennal response. So this suggests that olfactory capabilities of the two species are somehow determined by the sensory periphery. So I did a transcriptome approach where I compared the antennal transcriptomes of the two species in a parallel approach, and therefore I detected about 100 chemosensory genes in the antenna and compared the DNDS of this genome with genes that have nothing to do with chemoreception. And as you can see here, the DS is very similar between these two groups. However, DN is significantly higher in chemosensory genes than non-chemosensory genes. And this suggests rapid evolution of chemosensory genes between these two species. And I wanted to highlight one gene, OR41. This is an olfactory receptor, and it was the gene with the highest DNDS in the whole analysis. It was a DNDS of 7, which is extremely high. So I want to know whether these receptors play a role in reproductive isolation, and therefore I chose a population genomic approach. And I asked two specific questions. The first one is whether olfactory receptors show higher levels of divergent differentiation between and within than within species, and whether the divergence might be stronger in sympatric or allopathic population comparisons of these species. Therefore, I sampled populations throughout the whole distribution areas of the two species. 5 populations in total. Population 1 is allopathic for the glossal rudissima in squares. Population 2 and 3 are sympatric for both species. And populations 4 and 5 are allopathic for the glossal dilemma in circles. So the males I analyzed in 2 different approaches. I used genotyping by sequencing to randomly sample the genome for SNPs, and then targeted resequencing of chemosensory receptors that turned out to be interesting in the transcriptome approach. And using these 2 approaches, I can reconstruct a population structure to better understand the chemosensory gene evolution. So I'm showing you now a bunch of principal component analysis of SNPs that I detected using the genotyping by sequencing approach. And this is a set of nucleus SNPs here. And as you can see, there's not much differentiation at all between populations, which is maybe not that surprising because these 2 species are extremely close related and they seem to have very high population sizes. But if I concentrate on the 95th percentile of highest FSTs, the picture changes. The 2 species actually are distinct, but by the second principal component. And also, there seems to be some pattern of the geography, at least for Euglossia viridissima on the bottom. You can see that the 3 populations resemble how I collected them. In Euglossia dilemma, it is not quite that simple. So, although there seems to be a pattern of isolation by distance in this data set, there might also be something like with adaptation going on here. So, if I concentrate on the highest percentile of global FST, the highest FSTs, the pattern changes again, and here is where it gets exciting. I think 2 populations seem to be pulled away or pushed away from all the other populations. And these 2 populations are the sympatric Euglossia dilemma populations. They are different from all the other populations regardless of species status. So there seems to be some differentiation going on only in sympatric populations of Euglossia dilemma. And I also looked into this olfactory receptor 41 that turned out to be on a very high level of selection between these 2 species. And I sequenced multiple individuals in all populations of all species. And the picture that I get is pretty clear. I found a single allele for Euglossia viridissima is highly conserved throughout the whole area of distribution of the species, which is about 1,500 kilometers. And I used a branch site model to predict the yes, and it's predicted to be under strong purifying selective pressures. However, in Euglossid dilemma, the picture is different. I find two different alleles here. One allele I can only find in allophantry populations, and the second allele that is only found in sympatric populations. And it involves some strong positive selective pressures. So to wrap it up, the high FSD SNPs differentiate the sympatric Euglossid dilemma population from all other populations, regardless of species status. And the OR41 haplotypes correlate with sympatric allophantry properties in Euglossid dilemma, which is a very exciting result, I think. And so this suggests that something is going on in symmetry, but only in Euglossid dilemma, not Euglossid rhodizima. It seems to be differentiated from all the other populations. Also, I think that OR41 is a very good candidate for sensory differentiation between the two species, and because of these high patterns of positive selection, and also the two different alleles in Euglossid dilemma are pretty exciting. So, in the end, I think the results are consistent with reinforcement as a possible mechanism in the speciation process of these two species acting mainly on Euglossid dilemma. Yeah, and I want to thank a lot of people and funding sources and you for your attention.
Questions? Do you know what the OR41 recognizes, which compounds?
No, but we're working on
it. If OR41 gives function?
Yeah, it could be. But then I would, well, I don't know if I would expect two different alleles that I find in different populations. I actually look into different populations that are into more populations that are allophantry. And it seems to correlate pretty well with the connections of